Okay, it's all yours. You can start whenever you're ready. Whenever you
Okay,
go
I
ahead.
think we can give a couple more minutes. Rob, you can share your screen, okay?
Yep.
Yes. I'll give it a couple more minutes. Okay, so I will go ahead and introduce you as people connect. So Rob Dunn is professor in the Department of Applied Ecology at North Carolina State University. He obtained his PhD in ecology and evolution from the University of Connecticut in 2003, where he studied the recovery of tropical forests in Costa Rica, Peru, and Bolivia after clear cutting and use for traditional agriculture. I think this is very important and very pertinent, and you see how the biodiversity of the tropical forest can be correlated with the biodiversity of our own bodies. After he finished his PhD, he became a Fulbright Fellow and moved to Australia. I've heard several talks and by Rob, and he's an admirable science communicator, and not only has written several books, including The Wildlife of Our Bodies, that was published in 2011, to The Man Who Touched His Own Heart, that was 2015, but also his science essays have appeared in magazines like BBC Wildlife Magazine, Scientific American, Smithsonian Magazine, National Geographic. And he has actually known for efforts to involve the public as a citizen scientist in arthropod surveys and bacterial microbiota studies. And his projects in that area include, for example, the belly button biodiversity project and the mites that live on human faces, and the ants in the backyard, and the fungi and bacteria in houses. So thank you for joining us, Rob, and it's all yours.
Thanks, Andrea. So I'm going to start with a question today that I'm not going to answer, but I'd like you to start thinking about, which is this question of how will society and perhaps medicine in particular manage the relationship with beneficial microbes in the future? And we can think here broadly. We can think about gut microbes, skin microbes, agricultural microbes. What does this all look like in the year, let's say 2100? Or if we want to take a more specific version of this, in the year 2100, how do people acquire their beneficial microbes? And so we'll come back to this question, and I'll actually have a moment where you can sort of interact and provide some of your thoughts about what this future looks like. I won't offer my own opinion today about what the future will be, but instead some perspective consequences of different choices we make moving forward. And I have three main points today. And so if you're Zoom exhausted and want to look at the main points and then go and play with your cat, this would be the slide you need. The first point is that human microbiomes make more sense in evolutionarily comparative context. So thinking about our ancestors, thinking about our relatives, that allows us to make more sense of what we see in human bodies. The second is that as we consider the future, we need to develop ways of ensuring people can inherit the life that they need to keep them healthy and well. And the third, and this is just really the last teeny bit of the talk, is that a great deal of time is spent thinking about the far future of technology, but far less time about the far future of ecology, including our bodily ecology. And so Elon Musk spends a lot of time figuring out how he's going to make a house on Mars, but very little time thinking about the microbes that he's going to bring to Mars and what they'll do. And in the same way, this is also true when we plan our cities on Earth, we plan our hospitals. We're missing clear conversations about the intermediate and far future. In thinking about these things today, I'm going to draw on 3 publications. The first is this super fun paper I was able to write with a group of experts in primate evolution, baboon ecology, chimp ecology, hunter-gatherer ecology, and paleoanthropology, about what the microbiomes of our ancestors might have been like. And so I'll draw heavily on this paper, and I'll note when I'm doing so. And then the 2 other things I'll draw on are 2 recent books that both are, one came out this year, the next one's coming out in November. They didn't really get written at the same time, but COVID slowed one down and sped the other one up. But the book on the right, Delicious, is about the evolution of flavor in the context of our human ecology and human evolution, written with Monica Sanchez. And I'll draw on that a little bit today. This book's already available. I'll also draw, and actually more heavily, on the book that's coming out in November, A Natural History of the Future, which is really not about what will happen, but instead what biology tells us about what happens given particular scenarios that we might choose. First, though, I'm going to offer some insight into the comparative context and the value I think that comparative biology and thinking about our ancestry, thinking about our relatives gives us as we think about microbiomes. Before I get to that comparative context, I'm going to offer just a teeny bit of microbiome history, some of which comes from this book, but other bits of which come from elsewhere. And this is a reflection on how early really we started to understand that microbiomes can be important. I think it's tempting to think of microbiomes as being something we started studying 10 years ago. The truth is that by the 1890s, Joseph Leidy, for example, was already recognizing that that termites had what we would now call gut microbiomes. Here's this quote in the middle, I'll read quickly. So Leidy breaks open a termite and writes, "I observed brownish matter contained within the small intestine. It proved to be the semi-liquid food, but my astonishment was great to find it swarming with myriads of parasites, which indeed actually predominated over the real food in quantity. Repeated examinations showed that all individuals harbored the same world of parasites, wonderful in number, variety, and form." And Leidy would go on to write about these parasites that he called parasites in the old sense of the word, acknowledging that they may well be beneficial. And it wouldn't take long for people to realize they were beneficial. And when they did, the study of termites and termite microbiomes would go down the path that was extraordinarily holistic. The termites and the microbes were studied all at once and together as this whole that made most sense if you studied it as a whole, if you didn't atomize it into its pieces. Interestingly, at about the same time, importantly, for the termites, it was early on recognized that termites and their microbes were mutually unculturable. We still can't culture in the lab many of the microbes from the termites, and we also can't culture the termites without their microbes. And so this is a relationship that is mutual in many ways, and you can't really pull the pieces apart. At about the same time that it was being recognized that termites had microbiomes, it was also being recognized that humans had microbiomes. And there's a beautiful couple of studies by Escherich, writing in German originally. This is the translation. And this paper comes from 1885. And this paper already is recognizing the value of microbes to human babies. It's recognizing that some of those microbes are acquired from mothers, and it's recognized that the amount of microbes that the baby gets from the mothers may have effects on the baby's health. 1885. And so really very modern feeling. And so one thing that could have happened next in the study of humans would be that it would go like the study of termites. We would recognize that humans and our microbes are also mutually unculturable. But that's not what happened for the most part. For the most part, what happens is that the study of human microbes becomes one of counting, naming, and finding pathogens mostly, typically, well, in the absence of comparisons to other animals. And comparative research really becomes rare, as does holism. And so even though the termites and humans in this case are quite similar in many ways, they go down two very different routes with regard to study. Now, there were interesting exceptions, but even in the time, they felt like exceptions. And so there was beautiful work showing that you could culture beneficial Staphylococcus, inoculate newborn babies with it, use that to chase off the ancestors of MRSA, and that there was this sort of gardening of the microbiome that you could do if you thought about the body holistically. But these were kind of exceptions. For humans, the microbes were this secondary thing relative to the body, which was central. And then the other origin of these microbiome studies was really in farm animals. And in farm animals, from the very beginning, the studies were quite different and tended to be much more functional. The studies were about how do we keep farm animals healthy? How do we get them to make better meat, more milk? And so it was really a lot of the work very early on was about the functions of the microbes. And so if all these people were in the same room, each of these lenses offered something different to the study of microbiomes, but very rarely were these people all in the same room. And this would become more true through time. And as a result, we lost historically the holism of studying microbes and their hosts and their functions together. Termite biologists kept it, but nobody talked to termite biologists. They're always in the basement of their building. Not even the ant biologists like them. And so even though they had great insights, they were really kind of isolated. Microbiologists were often in separate departments, which tended to be have narrower functions. And people didn't compare different animal taxa. So we lost this holistic picture across animals or life more generally. One studies termites, one studies farm animals, one studies humans. They don't talk. This is a cartoon. Obviously, there are exceptions, but far fewer than one might imagine. And we lose that comparative context. And so I'll talk about these last two things today, and I'll do them in sort of reverse order. I'm going to start with the comparative context. In my lab and with my collaborators, and in the context of our paper on hominin microbiomes, we've thought a lot about hominin evolution and the context that it offers. And so I'm just going to quickly walk through some very quick case studies of places in which understanding not just human microbiomes, but also the microbiomes of our relatives, or even the morphology and physiology of our relatives puts humans in a different light. And so one of these contexts is the stomach. I'm using a crab-eating macaque here just as an example. If you look at textbooks on human physiology, and I just pulled this from a brand new textbook, just printed 2021, they'll typically say that the stomach temporarily stores food and also initiates digestion by churning and by secreting proteases and acid. This is what we teach students. Doesn't necessarily mean it's what we all believe, but something like this is sort of the status quo of understanding for humans. But Deanna Beasley, who's now at the University of Tennessee, Chattanooga, was looking at the literature on humans. And Deanna noticed something funny, which is that when she looked at the literature on non-humans, that the role of the stomach was described very differently, particularly with species with acidic stomachs. And so, for example, a recent paper suggested that it's been suggested that the vulture stomach acidity serves as a filter of potential pathogens. And so Deanna started to wonder, well, what if the stomach has this secondary role and sometimes a primary role of filtering the microbiome, filtering what gets to the intestines? And if that was the case, what she would expect to see is that species that eat more food that's more likely to have foodborne pathogens and it should have more acidic stomachs. And so Deanna actually did the first comparison of stomach pH that seems to have ever been published. And when she did, what she found was, in fact, that in general, things that eat more pathogens have much more acidic stomachs. And so you see in the far left here, the turkey vulture representing scavengers that have quite acidic stomachs, and facultative slightly less acidic stomachs. And then things that actually ferment in their foreguts have neutral stomachs. And so this is in line with what you would expect, actually, if there's an important role of the stomach, not just in digestion, but also in filtering pathogens. And this got even more interesting when we later zoomed in on primates and in looking at primates, one of the things that quickly stands out is that the pH of the human stomach is super, super weird. It's very acidic. It's far more like a vulture or a hyena than any existing primate. And then the other thing that we saw when we started to look at this is that there are very few data on stomach pH across species. And so hyenas are often talked about as having super acidic stomachs. We have never found a study of acidity in hyena stomachs, not a single one. Chimps are super interesting because the only data point we found suggested that chimp stomachs are neutral, like langur monkeys, which would be extraordinarily interesting. It's based on one captive chimp. That's all we know. And so here's this interesting suggestion that the stomach could be doing something quite different, and that this difference may have evolved relatively recently if chimps really have something like a neutral stomach. And importantly, this has clinical relevance. And so if this is all right, it raises the reality, the risk that any procedure or medicine that raises the pH of the stomach and makes it more neutral increases risk of pathogens surviving to reach the intestines. And in the broader sort of hologenomic perspective, that species with more acidic stomachs, including humans, may have a lower risk of pathogens given the same diet, but also more difficulty in reacquiring needed microbes. And so if your stomach is acidic as an adult, it may be far more difficult after taking antibiotics to get the microbes that you need than if your stomach were neutral. And so this is very much work in progress, but it changes our perspective on what the stomach might be doing, or one of the roles of the stomach. Another interesting sort of contextual comparative piece comes from the study of the size of the large intestines. And again, the textbooks haven't caught up with all that we're understanding about the role of the large intestines in humans. And so if you look at the textbook description of the large intestines, it's where you get the reabsorption of fluids and electrolytes and the storage of fecal matter before expulsion from the body. No mention of microbes. From a broader primate perspective, this is very surprising. It's also surprising in light of what we learned in the last decade. But in the broader primate perspective, we've known for a long time that the large intestine can play a very important role in metabolism. And so the question is, why is this not seeped into our standard textbooks for human biology? And explanations that's often offered relates to the relative size of the large intestine. Well, so in this case, the colon plus cecum plus surface of the stomach to the small intestine. And so the old story is that humans actually have a very small cecum plus colon compared to other primates. And so a very reduced potential role for microbiomes in helping to metabolize food. And so in general, the standard thought is that species at the left of this figure really don't rely on their microbes very much, and species at the right of the figure do. Now, obviously, we now know this is wrong, but this is sort of still the standard thinking underneath a lot of how we talk about humans and their exceptionalism. What became really intriguing, and also I would say that this is also the standard thinking around paleo diets. It's the standard thinking around the role of fire. And the argument here prehistorically is that what happens is that the descendants of Australopithecus africans invent fire, they begin to cook, they begin to process their food, calories become easier to obtain, and the microbiome becomes less necessary. But all of this depends upon how good the data are indicating where humans fall in this story. And so Amanda Hale, who you see at the top here, and Aaron McKinney got really interested in this a number of years ago. And they teamed up to look at human cadavers to see how much variation there is. Are humans really that far over to the left on this figure? And what they found when they looked at the literature is that the entirety of the data about the size of large intestines, small intestines in humans it's very sparse. And most of the studies of humans either have low sample size or produce or report no measure of the differences among individual humans. And so Amanda and Aaron decided to team up with a colleague at Duke to measure cadavers at Duke. And so they measured 37 cadavers just to get started. And in doing so, they found extraordinary variation in small intestine length and colon length and the ratio of the colon to small intestine. And it's not simple to map this back onto this figure, but more or less, it looks like that there are individual humans who are actually overlapping with chimps with regard to large intestine to small intestine ratios. And so our whole premise about humans being less microbially dependent as a function of the morphology of the gut may be partially flawed. And that only appears when you start to think about this comparatively. Another example, recent studies of huge numbers of samples of human microbiomes by the American Gut Project that I'm part of showed large differences when you look at different groups of people with very divergent lifestyles in different parts of the world. Don't need to dig into this too much, but each point is a sample. There are 11,000 samples here of 11,000 humans. And the individuals who are remote farmers or hunter-gatherers have quite different microbiomes, as you can see by the sort of spatial differences here from the broader US and British population. And so this has been known for a while. It's now been shown in many different contexts that there's this huge lifestyle effect on the microbiome. And so it's led to lots of studies of diet and lifestyle and how we alter the microbiome. But the other thing that study showed, and we replotted it in a recent paper we wrote, is that there's basically within the US and within England, no effect of geography or climate on microbiome. And so although there's this lifestyle effect, you can go to Kansas, you can go to New York, same basic microbiome that you're drawing from. There's a teeny effect of sex that partially shows up just as a function of the huge sample size. And so for the most part, no effect of geography or climate or sex. And so it's led to discussions that really emphasize lifestyle. But we recently studied chimps to figure out, well, what happens if we look at this same question? What's the effect of geography, of climate, and to some extent, maybe even genetics on microbiome? If we can study this question in a context in which we remove all the effects of global transit, of converging lifestyles and all of these other phenomena. And so we teamed up with folks at the Max Planck Institute in Leipzig, who have this amazing project in which they've studied chimps at basically every extant population across tropical Africa and collected feces at all those sites. And so we teamed up with all of these authors and we did our small piece of trying to contextualize the microbiome here. And so what we wanted to see was, is there an effect in chimps of geography, of climate? And if so, might this also suggest that maybe such effects were present in our ancestors? And then also, is there an effect of diet in the chimps? And what's cool about the chimps is that diet's actually decoupled from geography because it's actually most influenced by tool use. And so first and foremost, we did find a tiny impact of tool use on the microbiomes of the chimps. So the chimps that eat termites or ants have slightly different microbiomes than the chimps that don't. But we found a huge impact of geography and climate. And in fact, geography and climate together explain the vast majority of variation in chimpanzee microbiomes. And so this added context begs the question, was this also true prehistorically for our ancestors? And is the decoupling of geography and climate from microbiome an entirely new and strange phenomenon in humans? And we can't answer that yet, but it will be answerable. We've also worked a lot on skin, and skin microbiome has fascinated me since we first started working on belly buttons and trying to think about the ecology of the belly button as kind of a funny habitat that the public wanted to know about, but also that in many ways was kind of like a tropical rainforest. It was wet. It had interesting species. Most of the species weren't very well studied. And so we spent a while studying the skin surface, studying the glands, studying the bacteria and the fungi, and also the mites. The mites have been especially fun. When we started working on face mites on humans, Michelle Troutwine and I and other collaborators, we were actually the first ones to look genetically at these mites. And one of our collaborators actually found that the two mite species that live on humans, which were thought to be each other's closest relatives, appear to be 130 million years divergent. And so it's separate from the main points of the talk, but it's fascinating to me that we can find 130 million year divergent species on our skin, on our faces, that nobody really looked at. These are on every adult we've ever sampled. And so we've started to use these mites in a very specific way to reconstruct human history, in that the mites appear to record our geographic movements and then subsequent comings together. And so we do a lot of work on this. I won't talk about it too much more. It's super fun. I really enjoy it. But it hasn't yet told us a ton about our ancestral mites or our ancestral microbiome, so that we're going in that direction. But the other thing we've done is to work at skin microbes. And for skin microbes in general, there's tons of work in the 1940s, 1950s, 1960s that emphasizes the ecology of the skin in light of what is culturable on the skin, which is actually most of the species in the skin. Contrary to many recent publications, most of the things on the skin can be cultured. And the entirety of that work suggests that the normal condition for this skin is Staphylococcus, and that you have Staphylococcus and Corynebacterium and a few other taxa, Micrococcus, and that's sort of the normal condition. But there was basically no comparative work. And so we actually did the first study I'm aware of to compare human skin to the skin of other primates. And we wanted to see from this context, is that Staphylococcus story general? Is it just a human story? And how old might it be? And to do this, we focused on armpits. You see the weeping armpit. Weeping sounds bad. You know, it makes it the sweaty armpit of this gorilla. Gorillas, chimps, and humans, we all have apocrine glands in our armpits. And those glands do nothing other than to produce food for microbes, which is then metabolized, which then produces aromas. And in non-human primates, those aromas are thought of as being very important. They appear to be sexy in some contexts. They appear maybe to prevent pathogens in some contexts. They may be able to signal ill health. And gorillas, individual gorillas recognize other gorillas by their smells. But it's a very early literature that basically touches on humans not at all. The gorilla and chimp people decided not to talk about humans. They were bored by them. And so it's very not yet integrated. But we wanted to figure out, well, what are the differences in microbes? And how do we start with this? Teamed up with Julie Horvath Roth, an amazing scholar at the North Carolina Museum of Natural Sciences, and sampled humans, chimps, gorillas, baboons, and rhesus macaques. And first thing to notice is that the humans have fewer OTUs, fewer kinds of bacteria in this case than these other taxa. So that's interesting in and of itself. But the other thing that was really intriguing is that while humans indeed in armpits as elsewhere had tons of Staphylococcus and related species, that this was not true of the other species of primates. And those species tended to have more Corynebacterium, especially the chimps. And we know in humans that Corynebacterium is one of the species that is most likely to make your armpits stinky. And gorillas and chimps, surprise, surprise, have stinky armpits. And so, but the interesting question was when we started to look at this in more detail, some humans actually looked a lot like chimps or gorillas. And actually one zookeeper had an armpit that was indistinguishable from the gorillas. And so what's explaining that variation in humans? And so we started to zoom in on that. And I won't go into this too much today, but the key take home is that we did a controlled experiment that Julie Urban and Julie Horvath Roth led, where we had people wear antiperspirant, wear deodorant, and not use products. And then we had them stop and we studied their microbes. And what we found is that the people who had lots of Staphylococcus were either people who habitually used deodorant or antiperspirant or had applied it during the experiment. And so it's very clear that Staphylococcus abundance goes up with deodorant and antiperspirant use. There's more here, super interesting. It also appears to make people more attractive to mosquitoes. And so part of what's medically normal about Staphylococcus may be medically normal given antiperspirant and deodorant use. And so the picture changes. The other piece of this changes, thanks to Kate Coyle's work, is that we now know that there are individual genes. In fact, there's a single gene that's super important, ABCC11, that variants of that gene explain another 40% of the variation person to person in skin microbiome. And that made sense only in light of this broader context. And so the skin really changes when we put it back in context. It's true too for vaginal communities. So we work with Beth Archie and Elizabeth Miller. I gave a talk at Notre Dame, and we started talking about, and Deanna Beasley Skyped in. We started talking, and this is Skype because it's before Zoom. We started to talk about observations that Elizabeth had about vaginal communities of baboons. So baboon vaginal microbiomes, and they weren't acidic and they didn't have lactic acid bacteria, which led us to think, well, what does this look like more generally? And the assumption of human studies was that acid producing vaginal communities were and are the norm in mammals. But then when we looked, this isn't true at all. So actually the vaginal pH in the vast majority of mammals that are studied is actually very close to neutral. Chimps are sort of intermediate, and healthy humans are by far and away the most acidic. And if this is also matches the data on lactobacillus species. And so in humans, the average vaginal microbiome is 70% lactobacillus, very little in other mammals. And so there's something unusual about vaginal microbiomes of humans comparatively, and this hadn't been noticed. And what Liz and Elizabeth would go on to show is that part of what's happening here appears to be that there's a really high vaginal glycogen content in humans, especially humans who eat starchy diets. And that seems to favor lactobacillus, but this still hasn't been studied very well. And so there's a ton of opportunity for more work. And the fifth example here of comparative work is really stepping back a little bit to think not just about the bodily microbiome, but what I call the extended microbiome, the microbiome that exists outside of our body, and what really happens when our ancestors began to ferment food. And I will say that the literature on food fermentation is changing super rapidly with regard to our understanding of when food fermentation began. And so we argue in the book that there's actually quite compelling evidence that food fermentation may predate fire. And Katie Amato, one of our collaborators, and Liz Mallett, one of her collaborators, have actually documented that there are extant non-human primate species that actually appear to control fermentation. And so, for example, this capuchin monkey with its nice tonsure there, it goes into almendro trees, wild almendro trees, and knocks these big fruits down, and it can't eat them. They're too hard and too big to eat, but they will do this for hours. And what Liz Mallett noticed is that they would then come back 3 weeks later and eat them once they were fermented. And she's now documented this happening again and again, which looks like the control of fermentation. And so begs big questions about when do we begin to extend the microbiomes beyond our body? What does that relationship look like? How should that change how we think about our own body? And I say our own body because our work has actually shown that the interplay between the body microbiome and the food microbiome is much greater than I think we generally think. And this came in part from a study we did with Perato Center for Bread Flavor, where we had bakers from across Europe come together, Europe and Latin America, come together after having made the same sourdough starter using the same ingredients and the same rules in their home bakeries. They then came together, we sampled them, we sampled their sourdough starter. And what we were trying to figure out is, do they convey body microbes to their sourdough starter? And do those influence the flavor? And we saw that, yes, there is an effect. It's small, but it's there. And so you can say there's kind of a terroir of the baker in the bread. But the other thing that we saw, which was a total surprise, and here we are sniffing the baker's breads, is that the hands of the bakers were, even after they scrubbed, covered in lactobacillus bacteria. So the average study to date finds about 3% lactobacillus on hands of men, 6% on women. We were finding up to 70% lactobacillus on the baker's hands. And then the baker's hands were also covered with yeast. And so, yes, the bakers were changing the starter with their hands, but even more so, the starter was changing the bakers. And so this kind of extension, which is a new kind of comparative work, also changes how we think about our bodily microbiomes. The second part of this talk, which will go more quickly, is about the question of, given all of these microbiomes that we have on our bodies and then our extended ambit, how do we think moving forward about the inheritance of those microbiomes? How do we think about how we bring those not 2 years into the future, not one generation, but 100 years, 1,000 years, and what should we do? And here I return to the termite case, because for the termites, we know that when a founding queen, and I should say that this bulbous termite on the left, that's the queen, she's full of eggs. Termites, unlike ants, have a king and a queen. They're sort of British that way. Termites, unlike humans, the queen termite lays all the eggs for the entire colony. And so that's her hard work that she's swollen with there. But in termites, the queen, when she flies, carries with her the microbes that she and the entire colony will need. And the termite depends on that happening. And the termite also, in the colony, every time there's a molt, the termites have to reacquire their microbes. And so they feed each other. They do proctodial anus to mouth feeding to pass on those microbes every molt. And that sustains the colony. As we think forward, how do we account for this kind of dependence in our human societies? There's one way this happened in the past. What is it going to look like in the future? And I think in thinking about humans, we tend to think of ourselves as sort of holding this mobile of dependence, that species depend on us. But I think the reality is, from a broader perspective, we're actually more like at the bottom of this Calder mobile. And so how do we think about this? And you as a group know lots about the dependencies of humans on microbes. We see these things when our microbiomes go wrong. We see inflammatory skin problems such as rosacea and skin infections, MRSA, in the context of dysbiotic microbiomes. We see ear infections, cavities, bowel, Crohn's disease, nutritional deficiencies, obesity, poor mental health in the context of gut microbiome dysbiosis. In the context of having the wrong microbiomes in our homes, in our backyards, in our daily lives, we see other things: allergy, asthma, MS in some incarnations, and many other diseases associated with increased immune reactivity. So we know there are all these ways where if we don't carry forward with what we need, that things go wrong. And the added challenge is, if we think holistically about humans, we're very different from termites. Termites don't have to carry all the other things with them as they move to the future. They can depend on there being dead wood. They can depend on there being trees. They can depend on there being dirt. Humans, we consume tens of thousands of species, each of which is dependent on tens or more of other species. And so as we think about moving forward, we have to carry all of this with us. And I think that the International Space Station and the Mir, the Russian space station, are a good indication of how hard this is to actually think about, because we imagine these space stations to be a kind of future. And actually, when urban planners think about the far future, they often design things that look a lot like space stations, and often they have a bubble over the top. And so what does it look like when we think about space stations and the acquisition of these microbes? And I'll just say, as a quick aside here, that if we're going to control anything with regard to microbiomes, these are the places, right? You open the window, nothing comes in. Everything that goes up there is treated, it's controlled. The astronauts are screened. And so we might imagine if we can create a low life environment anywhere, this is it. That is not what's happened. Researchers have now on either the Mir or the ISS found hundreds of species of fungi, many of which degrade glass and metal. It was found on the Mir, tapeworms found on the International Space Station. So clearly we are unable to create environments where we just push the microbes out. And this is even clearer if you look at what the microbiome of the space station looks like. The microbiome bacterially of the space station most looks like a toilet seat. And so, here's this place we imagine as the future, and it's kind of a gross future. But the real, this is just an aside, the real reason I want to talk about this is because I think it begs the question of, if we think about a space station future, where do other species come from? And here I think it's very interesting to think about the astronauts. And so I had a chance to talk to Christina Koch recently and ask her about life on the space station, and hear Christina and Jessica Mir prepare for a spacewalk. And one of the things that was clear in that conversation is how very much connected to the life of Earth that the astronauts still are on the space station. And so partially this is emotional. Christina talked about that they often have plants on the space station that they grow to try out sort of student experiments. and that everybody's really excited to have the job of watering the plants, because when they water the plants, they get to smell the green of plants, and they get to smell the smells of the soil microbes, and they get to smell the earth. And so here's this way in which they still yearn for this connection to life in a broad sense. But then there are also the more mundane realities that where did Christina and Jessica get their microbiomes? Well, in all likelihood, from their mothers or from their hospital rooms, depending on whether they were born vaginal or C-section. They had some of their microbial exposures from dirt, from plants. And then all the food they eat was grown in the context of the microbes of farms on Earth. And more to the point, one of the key services provided by microbes and part of our extended microbiome is that of waste removal. And all the astronauts' waste comes back to Earth, probably not to be rolled by a dung beetle, but to be dealt with by earthly microbiomes. And so even the astronauts are very connected to life. And they're connected to the details of life, a reality that I was reminded of recently when I heard that it was discovered that on the space station, the plants on the space station appear to have a variety of plant pathogens that's causing the leaves to turn colors. And so now the space station is thinking about, should we be using probiotics on the space station to help the plants deal with the pathogens that we inadvertently introduced? And so we're connected. But that connection is so much harder for us as humans than it is for termites, because a termite queen, when she flies, needs only to carry her microbes. We must carry everything. And that everything has the added challenge that we still know very little. When I was a graduate student, one of the stories that really motivated me in tropical biology to go to those forests that Andrea mentioned at the beginning of my talk was the work of Terry Irwin, who went to the tropical forests of Panama as a humble beetle biologist to study ground beetles. And when he got there, he started to fog trees, the beetles fell, and he found that almost everything he saw was a new species. He invited his friends to look, and with different taxonomic expertise, and everything they saw was a new species. And so then he was invited by Peter Raven, who was at that time in an NSF panel, to estimate how many species there might be in a tropical forest. And Terry said, no way you could do this, but eventually he gave in, tried to do it. And he estimated the number of species in the tropical forest he studied based on the number of species of beetles that were only on one tree versus two trees. And then he estimated the global total of 30 million. And 30 million was exciting because only 1 million species of animals in total at the time had been named. And so that would mean that 29 out of 30 animal species were not yet named. Ferocious passive-aggressive fighting has revised this down to about 8 to 10 million, but still it's true that only about 1 out of every 10 species of animals on Earth is named. And so it's very hard to imagine that we know which species we need when 9 out of 10 are not named. But the even more remarkable thing about this is when we look at microbes, this gets ridiculously radical. And so Ken Losey and my collaborator Jay Lennon put out this estimate recently using scaling laws to predict the number of species of bacteria on Earth. And they estimated that there were 1 trillion species of bacteria. Now, there was a fair amount of debate in response to this estimate, but the truth is nobody knows, and nobody even has a good way of knowing. And even if they're off by 3 zeros, we still get 1 billion species of bacteria, and just 30,000 are named. And so all of this is to say that whatever it is we're going to depend on moving forward, we mostly don't know it yet. And so we need to carry with us into the future, into space, into wherever, the species in which we might someday depend, but don't yet depend, in addition to those that we know we depend on. And so this leads me to sort of closing the talk, which is to think about, how do we pull all this together to think about the far future? And so I think a lot about the year 2100, the year 20,000, the year 200,000. And for an ecologist, this is weird. People don't like that I do it. It's uncomfortable. And yet I think it's super important. And so I'm actually going to pause here for a second. I'll pause for 1 minute to let you all fill into the chat your views of the future for 2100, of how society will manage the human relationship with beneficial microbes in the future, or how will humans acquire their beneficial microbes in the future? What does this look like in 2100? And so I'm going to pause and take a drink while you fill this out for just Oh no, I'm still there. Good. Well, if you wondered what sound I would make if I thought that I just turned off the slides, that was it. Oh no. So I'll pause for a second to let you fill in your thoughts, and I'll go off camera and take a drink. And I think I can't see your thoughts at the same time that I'm looking at the screen share, so we can come back to them. Oh, here we go. Maybe I can. These are very interesting. Thanks for offering your thoughts here. I'll wait a few more minutes. Anybody else? Well, it's super interesting. So if you're, let's see what we do now. They will never leave us. Great. Well, keep typing, and I'll move forward a little bit. I think it's really interesting to pose these questions and to think about them, in part because my experience of institutions, and perhaps yours, is that institutions think about 10 years, businesses think about the next quarter. There's nobody really in charge of thinking about these longer intervals. And for example, when I've talked to city planners and architects, they often talk about a resistance from ecologists and evolutionary biologists with regard to their willingness to think about the far future and their willingness to think about the consequences of different human scenarios for life. But they also note that they will continue to build whether or not ecologists or evolutionary biologists are part of the conversation. And what is being built today will have a long legacy of consequences in the interactions between humans and the rest of the world. And there's an example that I think is fascinating in this regard and unites microbiomes and buildings. And it's the example that some of you might know of the Cloaca Maxima in Rome. And so the Cloaca Maxima in Rome was built as an open sewer by the Etruscans before the Romans. And it went through what would become ancient Rome, built around 600 or 700 before the Common Era. And already by 79, Pliny the Elder was super impressed that this Cloaca Maxima, which was eventually closed, still worked. And so he writes, in the streets above, massive blocks of stone are dragged along, and yet the tunnels do not cave. They are pounded by falling buildings. The ground is shaken by earth tremors, but in spite of it all, for 700 years, the channels have remained well nigh impregnable. And so the point here is somebody made a decision in 600 BCE about urban planning, then in 79 CE was still relevant. But even more impressive is in 1778, you still see the Cloaca Maxima exiting here. And in 1961, Lewis Mumford writes, with its record of continuous service for more than 2,500 years, that sewer structure proves that in planning of cities of low first cost did not necessarily denote economy. On these terms, the Cloaca Maxima has turned out to be one of the cheapest pieces of engineering on record. So somebody made a decision 2,500 years ago that's still relevant in 1961. And then if you look at it today, it is still working. And so we are making decisions that have every bit as long an impact on microbiomes today about how hospitals work, about how we store microbes, about how we do conservation, that have this same kind of impact. But what are people going to judge us on in 2,500 years? So the point is, the decisions we're making today, whether in hospitals or in city planning, are already the groundwork for a particular future. And the inertia of such groundwork is extraordinary. And some of what we do today will have impacts for centuries or longer. And our default is to build for the immediate, but we need to consider the longer term. And I will close here by noting that my book doesn't provide the answers with regard to microbiomes or anything else with regard to what we should do, but instead, around a whole suite of questions, offers insights into what life will do given different choices that we make. And here I'm elevating what we know about life, but also making clear what should be obvious, but so often is not, namely that regardless of the future we choose, that life will still be a protagonist. As Jose put it in his response, where was it, Jose? They will never leave us, that they will continue to interact with us, whether we choose to interact with them or not, that ongoing relationship is not going to be fully within our control. And so how do we take into account life's rules? And so it is a protagonist, life is a protagonist that's subject to physical and evolutionary and ecological rules rather than free will, and yet a protagonist all the same. And the interesting example that I'll just end on here is that in the Cloaca Maxima, since it was built, the crab that appeared in the Cloaca Maxima during Etruscan times and still lives there is now diverged into what appears to be a new species relative to the crabs in other water systems in Italy. And so we shape the world and the world responds, but the world also shapes us. And it's through an understanding of rules and generalities and comparisons and holism that we can make sense of all that. And so I will close there. Hopefully there are a few moments for some conversation. And thank you all very much. Let's see, hopefully there's still somebody here. It would have been awkward to have nobody. Yeah, great to see you.
Everybody's here. I think this was a fantastic, I think it's a big picture of what we do as researchers. Like many times you have, so you mentioned so many collaborators, and you have collaborators that work with different species and different environments. And so you put together this talk where you look like this is like such a broad picture and how everything is connected. So I think we have a few minutes for questions.
You know, I'll start off. For starter, this was absolutely amazing. I really enjoyed it. And Rob, thanks for thinking about this, because it's something I never thought about.
Oh, thank you.
Maybe we could, maybe I could pose a question to you. I mean, how would you answer the question? You wrote a book about this, and obviously have dwelled on this for years. I'm guessing that it's probably a hodgepodge of all of this, and the true answer is we really don't know. But how would you address your question?
That's a fair response. I think a first principle is that we need to be risk averse with regard to what we bring forward with us, and so what we conserve. And so we need great biobanks of microbiomes that appear to be healthy, but we also need to maintain those processes that are passing those microbiomes on. We also need to maintain those forests where the chimps are, who shed light on this story. And so part of what I think we need is to make sure we still have enough of the pieces that if we're wrong about what we understand today, that we can change our plan. And so that's one part I feel strongly about, that no doubt we're wrong about some pretty big pieces of this right now, but we won't know for a while. And if we lose what we need to go back, I think it puts us in in a bad position. I also think probably in practice, a good plan might be one that's conditional, that if a baby can acquire their microbiome via vaginal birth, that's probably will remain the best solution for a long time. But many many mothers can't have vaginal births, and so in a C-section, How do we think about the best microbes we can make sure the baby has? And so sort of these conditional responses. But I think we'll do the best job if those responses include conversations between clinicians, ecologists, traditional microbiologists, ethicists, and then we need a lot of people talking about these things, because very often if we just go decision to decision, we're led toward particular technologies, whether we think that's the best answer or not. It's a little bit of a cop-out answer, but I think it's an answer we're going to have to come to as a community. There needs to be a conversation about this rather than just what I think. But
I agree, and I do think it is incredibly important that before Aboriginal bacteria disappear in our as we deplete the ecosystems of the world, that many investigators are actually trying to preserve those communities, at least, as I've said, synthetically. So it'd be interesting to see, can that be done? And if you transfer that into a modern individual eating the crappy diet that we do today, how many will be preserved?
I also think that that's one of the, the real tricks of the simple capsule moving forward, as you're superimposing us in the chimps. The chimp microbiomes seem to be very specific to particular places and related to the history of the chimp, the genetics of the chimp, and what the chimp is eating. How do we think about a medical system where the microbiomes that a baby gets relate to its genetic background, its mother's diet, its potential diet? There are a lot of big questions there that I don't even know how we quite think about them.
Well, intriguing. Thank you so much.
Thank you.
In fact, so, well, no, first, if anybody else have another question.
I have a question. That was a great talk. I really enjoyed it. This may be diverging from the overall picture here, but how does being in space change the genomes or genomic integrity of microbiota? Is that known? I imagine they're subject to lots of radiation, just the way people are. Is that something that's been investigated?
Yeah, it's not a literature I know super well, but it does exist. And so there are definite changes. People have shown it both for fungi and bacteria. And there does seem to be some selection, and it's not clear whether it's via radiation or other factors for more extreme tolerance. And so the lineages that go up to space, when they come back down, seem to be more likely to be tolerant of more kinds of conditions. I don't know that literature well enough to comment on details, but it does exist. I mean, it's probably, 10 papers, not 100, but I can point you to some of them. Send me a follow-up email. You can find my email easily.
Thank you.
Yeah. Well, thank you so much, everybody. I hope you have a wonderful Thursday, and thank you for the invitation. And I'm here in the Triangle, so if any of this is worth following up more, let me know.
Thank you, Rob. Bye, everyone.
Goodbye. Stop recording.